Hi guys, Anna is here, and in this video we're going to discuss the process of action potential propagation. So please grab a piece of paper and so you can do your notes alongside with me. So the action potential propagation is the topic of the part of the nervous system series. Again, it's the upper six topic, which can be a little bit overcomplicated, and I'm here to help you simplify it. So to simplify this topic, we need to think of this topic in three components. So propagation is actually subdivided into three stages. But rather than subdividing it by stages, because this is probably how you have already been taught, I'm going to separate it slightly, divide it slightly differently and into the types of the channels. So you need to know of the sodium channels, sodium ion channels, which will be transported by the facilitated diffusion. Then you need to know of the second component, potassium channels, which are also be transporting potassium ions via facilitated diffusion. And then there'll be a third type of channels, or particularly a pump, which is called sodium potassium pump or exchanger, which will be involved in the active transport. The way to think about sodium ion channels is if we just draw like a little part of the membrane and we draw the protein channel itself, and we label the outside and the inside of the membrane. So the sodium ions will go into the inside of the cell by the process of influx or facilitated diffusion. Similar case will happen with potassium ion channels. Again, if we just draw part of the membrane, but the difference here is that potassium ions will exit the cell from the inside to the outside. So this will become important in a moment. And then the third type of channel is sodium potassium pump. And as a reminder, in this case, the ATP hydrolysis is used in order to pump out 3 sodium ions and to bring 2 potassium ions in. So guys, if you can learn these 3 types of channels, then we're set now to understand action potential propagation and the mechanism behind it. So the action potential propagation is going to occur across the neuron and especially along the axonal lengths, and is going to be subdivided into 3 to 4 stages. So stage number 1 is called depolarization. Stage number 2 is going to be repolarization. Stage number 3 is hyperpolarization, which is then going to bring us to stage number 4, which is the maintenance of resting potential. And to be honest with you guys, I think repolarization and hyperpolarization we're going to put together. Stage number 1 is the depolarization. So this is the arrival of the electrical impulse, action potential. So let's draw the section of the neuron or part of an axon. So if you guys just draw like 2 separate lines, which are going to represent the membranes of the axon. So, you know, the above area is the outside, and then the middle area is the inside of the neuron. And what we're going to do now, we're going to bring those 3 components that we separated by in the first slide, and we're going to draw them together. So let's color code it as well. So this is going to be a sodium channel in blue. Again, guys, you're welcome to use any colored pens here. Then we're going to have a second protein channel, and this is going to be a potassium channel. And what you can notice is that I've drawn them slightly different. So the sodium channel is here going to be opened, and then the potassium channels are going to be closed. And then the third component, sodium potassium pump, is also going to be closed and not going to do any transport in this case. Okay, so if you guys can separate into 3 components, so obviously there are going to be so many more channels here, but we can't draw them here. We just drew and separated them by type. So in this case, as you can see, that the sodium channel, so the sodium protein channels, they're voltage gated here. It means they open due to the receival of the electrical impulse. And as you can see, the sodium ions, they enter by the process of the facilitated diffusion. So we can call them we can describe it as an influx of sodium ions across the membrane into the axon. And that means, guys, that it will go from resting potential from about -70 millivolts to about plus 40 millivolts, but don't worry about these numbers. I'm going to describe them later on the graph as well. But so what it means is that the membrane potential, or the voltage against the membrane, has increased due to the influx of sodium ion channels. And the more sodium voltage gated channels open, the more they will activate their neighbors, and there will be more of an influx of sodium ions. Okay, this brings us to the second stage of action potential propagation, called repolarization. So again, let's draw three different components. As you can already notice from my diagram, I've closed the voltage gated sodium channels. The potassium channels, voltage gated as well, they will be now actually opened. And then the third component of sodium potassium pump, it will still be closed. Okay. And this kind of should bring the memory back from one of my earlier slides, where what was the mechanism of potassium channels? Well, it's basically the potassium channels, they leave the axon by the process of the facilitated diffusion. When something exits the cell, we can call it as an efflux of potassium ions. And the key thing here is, guys, if you have the positively charged ions leaving the cell, then the inside of the membrane, so the membrane potential, is going to become less positive. Okay, and in this case, we actually can start and join the repolarization and hyperpolarization together, because it basically dependent on the efflux of potassium channels, as I will represent on the action potential graph shortly. This brings me to the final stage of the action potential propagation, which is the maintenance of resting potential. And as you guys can guess, that the third component that we've drawn before is actually now going to be working, which is the sodium potassium pump. So the sodium channels, ion channels, will be closed. The potassium ion channels will also be closed. And remember, guys, they're both voltage gated, meaning that they respond into the changes of the membrane potential. And then the third component, sodium potassium pump, will be working now, in which case the ATP hydrolysis is used to pump 3 sodium ions out and to bring 2 potassium ions back into the axon. And if that happens over the time, you can imagine 3 positive ions are leaving and 2 positive ions are entering, meaning that over time there'll be -1 of the charge happening. So therefore that maintains it at the negative. Right, let's now bring all these processes to represent them on the action potential graph, which is commonly comes up on exams. So I'm just going to draw this not to scale, but let's put -70 millivolts as a resting potential plus 40 millivolts. And I'm just going to draw this shape, which I want you guys to replicate and copy over to the notes. The region that goes from -70 to +40 represents depolarization. Then the region that goes down represents repolarization. That undershoot where it becomes more negative represents hyperpolarization. So it's too many potassium ions are transported out. And then the shape of the graph returns back to the resting potential, which is around -70 millivolts. And that is achieved by the sodium potassium pump. Okay, guys, so I hope you found this video very useful. Thank you for all your continued support. Please press the likes and subscribe to my channel so I can deliver more of those videos to you. If there are some certain topics that you want me to do ASAP, then please do leave the comment below or any questions and I will respond to them. Thank you so much and see you in my next video. Goodbye.